one of the core things to start off with around this is that Parkinson's disease as a movement disorder is highly heterogeneous in its presentation, meaning that one person with the disease will present a very different set of symptoms to another. Although Parkinson's is seen most primarily as a motor disorder, it can present with a number of secondary symptoms which are much more common in Parkinson's compared to other populations, including things like depression, anxiety, hallucinations, dementia, apathy, sleep disorder. The list goes on. But broadly, we kind of wanted to ask, what can we tell from this heterogeneity in how they present, and whether one person presents with certain symptoms or doesn't, whether there's some sort of underlying information being carried there about the pathological processes around Parkinson's disease. So we look at this through the lens of epigenetic data. Epigenetics, in a nutshell, refers to a set of molecular mechanisms which alter the way in which the underlying genome is expressed. And the idea there being that it can modulate overall genetic risk in how it is expressed in individual phenotypes. So we believe that that might carry some association to how different people present with the disease, and sort of use that to probe further as to whether we could pick up epigenetic associations with differential symptom presentation in Parkinson's disease. So we looked specifically at DNA-methylation, which is an epigenetic modification to the DNA nucleotide sequence. It's a chemical tag that gets added to the DNA molecule, and it has been shown to sort of up and down regulate specific genes sort of around the vicinity of this methylation and also sort of further on. And it's been previously associated with neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease, but hasn't really been looked at in too much detail in terms of like subsymptom presentation within the disease. So we sort of looked specifically at that question, and using a cohort of 97 Parkinson's patients generated from collaborators in Cardiff University, looked at DNA methylation genome-wide across hundreds of thousands of different sites in association with 6 common secondary symptoms that you see within Parkinson's disease. What we tested is actually not each individual site. We aggregated hundreds of thousands of sites to smaller networks of methylation and tested their correlation with these secondary symptoms in 3 different brain regions relevant to PD neuropathology development, these being the substantia nigra, the caudate nucleus, and the frontal cortex. What we find principally as sort of a key finding is that we detected DNA methylation networks specifically within the substantia nigra that shows significant correlation to depressive symptoms within Parkinson's disease. And when we follow up sort of interrogating this network, we find that it's highly enriched for terms relevant to neuronal and synaptic processes. And then taking reference single nucleotide RNA-seq data that's publicly available from previous publications, we find that the expression of these genes annotated to this network are also highly enriched for expression, specifically within dopaminergic and non-dopaminergic subtypes of the substantia nigra. So this sort of principal finding that we present here and that we sort of are highlighting indicates that neuronal changes within the substantia nigra, which is more principally sort of previously associated with motor symptom development within Parkinson's disease, actually might have a role in the development of these secondary symptoms, specifically depression. And this sort of matches up with some previous neuropathological findings. So what's been shown for the substantia nigra when people have looked neuropathologically at the region in relation to depression and PD and Lewy body dementia is that depressed patients have a higher burden of alpha-synuclein pathology within this region, and there's some evidence that there's greater neurodegenerative effects here. So what we highlight from this study is potential cell types involved with this and potential genes that might be either driving or a consequence of this process. The implications of this study, I guess, are twofold. One is that DNA methylation does appear to pick up and explain a level of this heterogeneity that we see within disease, in particular in relation to specific subsymptoms within Lewy body disease and Parkinson's disease specifically. But what we see is that these effects are highly brain region specific. DNA methylation and epigenetic changes aren't like the genome. They're not fixed at birth in the same way. They change over your lifetime, and therefore it sort of makes sense that they would be specific to certain brain regions within the disease. So I guess the implications from that is that you need to be asking very specific questions and looking at very specific areas when looking at epigenetic data. In terms of how we're like following up on this, we have sort of 2 follow-up studies that we're sort of interested in. One is a larger cohort that we generated internally, which is looking more specifically into cognitive symptoms, both within PD and DLB, and looking at cortical regions relevant to the development of pathology and cognitive symptom development. That's also actually being presented at AUK by my colleague, Dr. Jenny Im. We're also looking to leverage that cohort and some other available DNA methylation cohorts that are available out there to look at whether we can do data-driven subtyping of disease using DNA methylation data to pick up disease-relevant clusters of methylation that sort of differentiate patients, and whether they can be used to kind of explain further heterogeneity within the disease.